again, this is now lecture 13 in virtual tissues modeling. As always, I need to remind you that things are being live streamed and recorded. I hope those lectures are sometimes useful. I know I don't get them edited all the time immediately, but you have that material available. Today, we're going to be talking about antimony models again and their integration with multicellular organization. So we'll go over a little bit of antimony syntax again from last time, since that's something that we need to remind ourselves of. Again, also, there was that one-page cheat sheet that has the entire syntax. Antimony syntax is very simple. And then we may briefly review some of the exercises we did at the end of class last time. And then I'd like to get into more sophisticated models. There's a delta notch patterning exercise, which is a classic one, which we always do. And then I'd also like to do, we may skip one or two of the detailed exercises, but I'd like to do the cell cycle model of cell growth, which is a classic model, which couples the subcellular model and the volume term in the simulation. And then, depending on how much time we have, either it'll be in class or homework problem, which is Zartman's model of calcium waves, which in many ways is very similar to the model that we were working with on delta notch. And this really, that Zartman model, and actually the delta notch model as well, are things that are currently used in research. I mean, these are based on published models, but the things they're based on are published in the past year or two. They're recent, and they're currently used by people for contemporary research. So we're getting to the point now in the semester where you have enough tools to be able to do modeling that is in fact research relevant. So I'd like to quickly just ask people where they are with their project. I know, Emma, are you and Pedro still working together?
We are, but we're also working with Alex. However, we need to touch base and get back into the loop because after spring break, at least unless they can meet.
Okay. Pedro, did you have anything you wanted to add?
No.
No. Okay. And I guess Santoshi and William, you've been meeting together and you want to have a Zoom this week, right?
Yeah.
Yes, sir.
Okay. So we'll do that. We'll get that set up for you. I wanted to give people the opportunity to ask any questions they might have about the homework assignments. If there's anything specific that you'd like to go over now, happy to do that. Also, again, if you think about something now that you want to bring up after the break in the middle of the class, we can talk about it then too. Kyle, go ahead.
Yeah, I just have a request for fellow classmates. I'm going through all the course materials and videos with the idea of making little knowledge objects of the items we learn in the course. It's quite a big endeavor, but we're working on some automated ways to do it. If any of you have a particular homework assignment that you're happy with, that you would like to donate to be a part of a knowledge object that somebody could go look at and figure out something from it as an example of a code or model that you're particularly happy with, I'd love to share that as part of the knowledge base. Of course, we got to check with James first and figure it out, but I'm doing that kind of stuff right now, making a knowledge base that would allow you to get up to speed faster on CompuCell 3D and particular use cases. And I'm really focusing on the easy early stuff first, but try to increase people's velocity to productive models, reduce friction to learning this stuff, as well as take whatever skills you have that you bring to the course and leverage the ones you have and fill in the gaps, because I noticed there was a lot of gaps in my skills. I had some of the software, I didn't have some of the multicellular stuff. And so we're trying to build out a knowledge base. So I don't know if we could get James' endorsement, or maybe your projects are the ones that make sense to put in there because they cover a lot of things. But I just wanted to put that on the table. Thank you for indulging me.
Thank you, Kyle. I mean, there have been requests for solution sets, and there's always a little bit of an issue with solution sets because the same problems get assigned year after year.
Yeah, I know.
But I don't in principle have a problem with people looking at old material as long as they actually do the problems themselves. As a hint, if you look at the videos from previous years, you'll find that a lot of the problems are actually done in them. So if you want solution sets, in particular for problems that classically, when people handed in the homework, I would look at what people had problems with, and then I'd spend the first half hour of each lecture going over the problems from the previous class that people had had issues with. And so actually, if you look at the previous videos, you may find that some of the problems that you are being assigned or worked in those videos.
Yeah, and I'll go back and use those too. I just wanted the birth parents, if they're interested in taking their little fledgling models and sharing them, we would be happy to annotate and work on them and make them into more robust learning tools.
But if they're homework assignments where you think I did a great job and you're willing to share it, that would be great too. So thank you, Kyle. That's a good point. All right. So last time we introduced the idea of biological networks, the multiple kinds of biological network, chemical reaction or metabolic networks, signaling networks, regulatory networks, PBPK or pharmacokinetic networks. They all have the same mathematical structure, which is a series of first-order rate equations. Those rate equations could be represented either as ordinary differential equations or as stochastic differential equations. They can also actually be represented as Boolean networks as well. In this class, we're not going to have the time to do the stochastic representations or the Boolean network representations. Suffice it to say that if you're interested in that, you could do it. CompuCell supports it, and if you want to learn about it, a good place to start is Herbert's Systems Biology Introduction to Pathway Modeling textbook, although there are many, many, many good textbooks. Uri alone has a wonderful textbook. There are many good textbooks on network modeling. They all have rather different flavors. Some of them are more biological, some of them are more mathematical. They cover different examples. It's a big field. I really think that Herbert's Read the Docs and the Quick Start Guide for Antimony are excellent. Really walks you through everything you need in very few exercises, and all the demos in his Read the Docs have executable code that you can try out. There's also that one-page Antimony to Learn cheat sheet. Again, the reason we use Antimony is mainly because it's so simple to write the models. Its downside is that its error messages are pretty non-informative because of the fact that it's parsed as a string. And so when you have typos, debugging is a little bit more complicated. So on the one hand, you have the simplicity of the modeling language. On the other hand, when you do make a mistake, it's a little bit harder to find. So the basic concept that we're dealing with are rates of change. In this case, we're writing a chemical reaction, simple chemical reaction, A plus B goes to C. That implements three rate laws, one for the concentration of A, one for B, and one for C. We typically write the velocity of reaction V. It's called V sub F because it's the forward rate of reaction. In principle, C could go back to A and B. Biochemistry, molecular reactions are almost always reversible. although we often ignore the reverse rate. If we don't know what that rate law is, we can't do anything more. Very often we will assume the law of mass action if we're talking about chemistry. If we're talking about gene regulation, then we typically use what are called Hill functions. We'll talk about those in a little bit. But law of mass action says that the rate of reaction is proportional to the concentration of each component raised to its stoichiometry. If I have 2 molecules of A plus 1 of B combining, then it would be A squared times B would be the rate. And the reason for that is a very simple one. If I want 2 molecules to react, they have to come in contact with each other. If the densities are low, the probability of contact between 2 molecules is proportional to the local concentrations of each one. If I need 3 molecules, then I need concentration A times concentration B times concentration C. And if I need 2 molecules of A and B, it's the same thing. It's A times B, which is why you get the exponents. And so depending on what you're doing, these chemical reaction models can be very compact because that one equation gives you 3 ODEs. Sometimes it's the other way around, where you have 1 ODE, you have to write 3 reactions to implement it. Depends a little bit on the problem. And so we did have last time a demo that would run antimony models inside of CompuCell. You certainly don't have to run them inside of CompuCell. Antimony and tellurium are Python packages on their own. But we wrote this little demo that allows you to type an antimony model and plot the time series. And in that, you had a place where you typed the antimony model just as a string. It's just a Python string. And then you defined which variables you wanted to plot, and it'll plot that out for you in time. Now, if you want to adjust the time scales and do things that are a little fancier, then you have to edit the code a little bit more, but this was designed as an introduction. We began to talk about how to connect CompuCell cells to the antimony model, and effectively, as is usual in CompuCell, we use CompuCell as Python dictionaries as ways of connection. And so if we load a model into a particular cell, then we reference cell.sbml.modelName and then the name of the variable or parameter that we want to reference. As I say, the fact that we say SBML is a bit unfortunate. It should be something like network. But the first network modeling language we supported was SBML. And so we're stuck a little bit with the SBML name. There's also a global ability to load the model globally instead of into cells. And in that case, it would be self.sbml.modelName to reference. The one time this is going to be a little bit awkward is if we want to use tracking fields. And so we'd have to copy from cell.sbml.modelName value to cell.dict value to display. You can't define a tracking field directly on the SBML dictionaries. So to remind you of antimony syntax, everything is inside of a string. In Python, if you want to be able to have carriage returns inside of a string, you need to use triple quotes. You can use either single triple quotes or double triple quotes, or triple double quotes, I guess is correct. Everything inside the string. And so you can put the carriage returns are decorative effectively. There are 2 basic kinds of comment. Actually, there's also the hash as well as the double slash for comments. That was added after this slide was made, which tells you it's an old slide. Semicolon can be used instead of a new line character. You can tell that antimony inherits from C++ in terms of its syntax a little bit. The basic concept again is a rate equation, something, arrow, something, semicolon, rate, semicolon. If you want to name something in antimony, you use a colon. So E1 colon T goes to I1, semicolon, beta times T times V, creates a rate law that T goes to I1 at a rate beta times T times V. One thing that you have to watch out for is you can't have multiple rate laws with the same name. So if I had E1 colon something, and then I had another line that was E1 colon something, that won't work. The other thing you have to watch out for is that there are reserved words in antimony. Sometimes they're obvious, like don't call a variable time, because time is a reserved word. Some of them are less obvious, like gamma is a reserved word. And it won't throw an informative error. If you do that, it'll just fail. And so if you're going to use common words like beta and gamma, I recommend putting a number in the name, beta 1, gamma 1. Then you won't clobber the reserved words. Okay. If you want to have complicated chemical reactions, which we don't do so much in this class, but if you were doing something like metabolic networks, you'd need that. You can write those with explicitly. If you have 3 molecules of A and 5 of B going to A and 2 C's and 9 D, you type it more or less the way you write it. You don't use a star for multiplication in this case. And antimony will parse that properly. It's now possible, although it's not something I've used, but in the latest releases of tellurium antimony, you can have the stoichiometry be a variable. So you could have number times number A, and number will parse, and you can have a variable stoichiometry. It used to be, that was a little bit tricky to do, but now it's possible. Notice that the exponentiation operator in antimony inherits from C++. It's the caret rather than the star star. So that's an easy mistake to make, because if you're typing Python and you're using double star for exponentiation, it's tempting to do the same thing in antimony. Okay, so that one equation there gives 4 rate equations for the different species. If I just want to write a simple ordinary differential equation, I can do that with nothing goes to the item on the right. There's a question, does anybody have a link to the list of operators in, you mean operators in XML?
Stop.
You mean for, okay, so, I mean, to some extent, that's going to depend on the parser you're using. So it's not going to be a generic thing. As I've said, one of the issues in the CompuCell XML is that in general the parameters are not parsed as mathematical expressions. Now, there are places where you know that they are. For example, when you write the initial conditions for a field, that's parsed as a mathematical expression. But most of the parameters specifications are not parsed as expressions. Might be something that would be nice to add to it as a parser. Okay. So if I want to write, there's a source of A, at a rate K, then dangling arrow, nothing goes to A, semicolon gives me the equation dA by dt equals whatever I put after the semicolon. Similarly, if I write A goes to nothing, then that gives me dA by dt equals the same rate with a minus sign in front of it. So again, there's a different biological concept, but it maps to the same mathematical form. You can specify ODEs directly. It's equivalent to writing nothing goes to A. At least functionally it is. T prime equals beta times T times V will give you dt by dt equals beta times T times V. That's functionally equivalent to writing E1 colon arrow goes to T semicolon beta times T times V. It's really a question of what you prefer to write and how you want to write it. All parameter values need to be specified. Tellurium will croak if you don't specify the initial the parameter values. I recommend commenting and giving the units of the parameters and a comment about what they mean. Variables don't need to be specified. If you don't specify them, they'll be given 0 initial values, but it's always good practice to give the initial values and also, again, their units and a comment on what they mean. Now, having reminded ourselves of the syntax, I want to jump into the problem of how we're going to integrate these network models with CompuCell. And in general, we can have quite complicated network models running inside each cell. We could, in principle, have the parameter values in each network model in each cell be different. We'll get to that actually today. You could, in principle, have different network models running inside of different cells. That's also possible. You could have multiple networks models running inside a single cell. So there are a lot of possibilities. This is the
that
cell
was
site.
what TJ was talking about today in his session. He couldn't get it animated, but the prior slide.
Right. Yeah, that's sort of our default slide showing what CompuCell can do. It's pretty old, but it's showing the different levels of simulation, the macro, mechanical, and the fields, and then the subcellular networks. In a sense, it's a nice demo. This is also a very old simulation. I hope we can do this exercise today in class of cell cycle. And the one we're definitely going to do in class is this delta notch model, where we have signaling between neighboring cells, which leads to this pattern that's very typical of developmental biology. That's a cross-inhibitory signaling pattern. Okay. So if we're going to specify antimony models, we always are going to have to do the same, essentially the same workflow. We're going to have to write the antimony string. We're going to have to load that antimony string as a network into either the whole simulation as a global network or into individual cells, one or more individual cells. We're going to have to time step the network models. We're going to have to be able to read and write their values and parameters, because if we can't access them, we don't do anything. And then very often we will have to couple them with something about the cell. If the networks run but don't do anything, it's not so exciting. And so typically we'll either change values in the networks from the CompuCell Python or read from the networks and modify the behaviors of the cells. And then we have the ability to unload network models, which we're not going to use in this class. But if you have very large models that are slow, it's often convenient to be able to unload them. So again, we're going to define the antimony model. It'll go in a string. You need to put it somewhere where it's accessible in your Python, or it could be in a file and read from a file too. And if you're going to use multiple models, then you have to have multiple strings or multiple sources. One thing is that you have to keep track of the conversion between antimony time units and CompuCell Monte Carlo step time units. I have a couple of slides which are hidden, which go into some best practice on that, but I'm not going to belabor that today. There are a variety of options in the loading the model. Again, you can load a global model. You can tell it, CompuCell, to load the same model into all cells. You can load it into all cells of a given type. You can load it into a particular cell ID and loop over the cells to do that one at a time. Those are all possibilities. So it's up to you to decide how you want to do the loads. So the syntax is going to be add antimony model, and then depending on which of these you're using, to cell, that'll load it into a cell using the handle of the cell. Add antimony model to cell ID. If you know it's cell number 19, then do that. Add antimony model to cell types. We'll add it to all cells of that type or that list of types. And if you want to add it to the system as a whole, it's add free-floating antimony model. You have a parameter in the optional parameter in the load, which is step size, and that specifies the time unit conversion between antimony and Monte Carlo steps. You have the option also of loading from a file, in which case you would do model underscore file equals and then the path to the file that you want to load. During the load, you can overwrite pre-existing values of variables or parameters. And so you can use the command initial conditions, and that will allow you to overwrite values that you want, or you can load the model as it is, then loop through the cells, loop through the dictionaries, and write the values that you want. Again, everything is accessible as a dictionary entry, cell.spml, name of the model that you defined, and then quote the name of the variable or the parameter. You don't need to use a get or a set function. They're there. We don't use it that often, but there is an option of doing result equals self.spml.modelname.values, and that'll return all of the values at once. And if you want to see how that's structured, I recommend doing it and trying a dir on it to see how to read out the information from it. So at the end of last time, we had done a little exercise, and I'd like people to get that going again. Make sure everybody's up to where we were before. And I guess I have to somehow the formats on some of these slides got clobbered from yesterday to today. So I apologize. I can fix that while we're working on it. So I'd like you to create. You should have it from last week. If not, create a simulation with 2 cell types. You could set the cell volume to 125. Doesn't really matter. Create a circle of cells. And then we're going to load an antimony model into the cells. We're going to try some different options. We're going to add 2 attributes A, B to the cell dictionary so we can display their values using a tracking field. And then we're going to set the values of cell attributes from the antimony model. And I'll walk you through that. So the first step, here's the model we want to use. Do people have the model from last week? So if you haven't done this before, use Wizard to just generate 2 cell types in a cell lattice with blob initializer, an initial target area of 125. And so I'll give people a minute to do that. And then once you've done that, all of the other steps we'll walk through one at a time. So you probably have a contact energy, a volume constraint, Bob initializer in the wizard. There's also a version of the code in the demos from last week. Hayden, would you be willing to put the link to that in the chat for me? I can look it up
Yeah, I'll see
too.
if I can find it.
For everybody else, if you could get this running, that would be good. William, do you have it working? So do you have your code from last week?
I'm pulling that out, but I don't think I got the two thing going on for right now.
Okay, Santoshi. Again, I'll walk you through what you need to do for the antimony. For the moment, all you need to do, if you don't, is to create the initial condition and the cell attributes in Wizard if you're going to do it from scratch.
A and B would be the concentrations of A and B.
Right. I'll walk you through that. Don't worry about that. This all really only focused now on step number one, which is to use Wizard to create a simulation which has two cell types with volume energy, contact energy, and use blob initializer to create a circle of cells. That's all you do. Okay, good. So, okay. So William and Santoshi, do you have those done or not? William?
Can you hear me? Oh yeah. Give me a
Yes.
sec. Like I'm reopening my files. I'm trying to find the recent one I opened.
You could just generate it from new too. It only takes a minute.
Oh, okay. I can share my screen.
Okay.
So I just opened choose a new project, right?
So
And we just keep all these the same, right?
yes, just hit blob initialize.
Okay.
Okay, that's fine. Sorry, Emma, you won't see this because it's the way the screen's set up is. Okay, create two cell types. That's fine. Next, you don't need fields. You need contact energy, volume flex. And that's it.
Okay.
Okay.
Got it.
Okay, good. Santoshi, what about you?
I too just generated the same simulations. So
I'm sorry, what did you just say?
yeah, I too just generated the same simulation. So
Okay, you're at the same point, so you've got
yeah.
that. Okay, good. Okay, so we've all got that. So the next thing we're going to do is write a very, very simple antimony model. This isn't going to do anything very interesting. But we need to have some models just for practice. And so you can put this at the top of your steppables file. After the initial loads, but before all of the other declarations and classes. Okay, and you don't need the comments. You can just say model equals triple quotes J0 colon A goes to B semicolon K1 times A semicolon. You don't need the equations, parameters, initial conditions, comments to make it faster. The one thing you do have to be careful of is that there is a difference between a colon and a semicolon. And if you get one of those wrong, you will fail with unhelpful errors, which I think we found last week. So I'll give people a minute or two to type that and give me a thumbs up when you have that. Okay, Emma says it's okay, William.
Shit. Oh, I'm checking the commas and semicolons. I think I should be fine.
Okay, and Santoshi? Santoshi, is that
Yes, sir, I did.
you've typed this
Yes.
now? Okay,
Yes.
fine. So now in the start function, we are going to load the model into cells. And to begin with, let's just load it into all cells. And as always, we'll do a for cell and self.cell list. And then we'll do self.add antimony model to cell. And then model string equals whatever you called your string. Give the model a name. Cell equals cell. And then we'll make the step size 1. Okay, so use the last one there. And you don't have to type all of that by hand. Use the wizard to, in the SBML section of the wizard, to give you the syntax for that. The syntax that you're going to get from wizard will not be exactly what you need to type here, but it'll be pretty close. So you can paste that in and then edit. So I'll give people a minute to do that. Give me a thumbs up when you have that working. And then in the step function, you'll do a time step, which is self.timestep underscore SBML, open close parentheses. Again, that's available in the wizard. And I recommend always, every time you add one of these things, try running the simulation. If it crashes, you have probably a typo. Did that work for people? Emma?
Previous slide.
Yep.
Unless I read it incorrectly, the method was add antimony model to cell.
Yeah.
And then the slide is just add antimony to cell. And I think that
That's a typo. Use the syntax that wizard gives you. You tell me what it should be.
add to cell.
Not add antibody model to cell. So I'll fix it.
Now it's working.
Which is correct? This one?
This.
Okay, then I'll fix it in the other one. Okay, thank you. In general, I tend to use these were cut and pasted from simulation, so they should be right. I tend to use wizard, and so I don't always remember the exact names of the functions. So it's possible that I typed it wrong. And then if you can time step the model in the step function.
Yes. So when I'm running it, it looks like A and B are the same. Although
Yeah, they're
it's
not going to do
different.
anything interesting. This is a very boring model. It's really just to do the mechanics of getting models loaded and running. Santoshi, why don't you screen share what you've got? Okay, let's look at your code. So what are you doing? What are you doing here? Santoshi, what are you trying to do?
Like loading into all the cells. I'm sorry, just to a cell, I guess.
Okay. Do you know there's a cell ID 20? It's okay if you do. In any case, line 31, you're missing an underscore. Okay. Okay, that should work. That will load it into one cell. Assuming you have at least 20 cells. That's fine. Okay, try running that. Save it and run it. William, please, Kyle, please keep working on that while we're doing it. Yeah, we can't have a space. Model name.
Okay.
So I understand that a lot of people haven't used Python, but it is a good idea to remind yourself of basic Python, so that you can catch these things. It's easy to. Python is not forgiving of typos. Okay, try that. William, how's it going?
I typed the same thing as the slides, but it doesn't work.
Okay,
Yeah, it's like totally the same. I don't know why it doesn't work.
okay. Well, we'll take a look as soon as we get Santoshi.
Okay, yeah, sure.
Uh,
Okay. You called it KA, KB, not K1, K2.
Okay, sorry.
Okay. Okay, fine. So, William, do you want to screen share and we can
Yeah,
see
sure.
whether we can help you out?
So these are my code. And I don't know what.
No, well, you can only do one of those. You don't want to do all of them.
You mean like this part?
Yes?
So I just like delete all of these and it will
No?
work?
No,
No?
you only use one. What do they do?
I
What
thought
do
it's
they
like
do?
it's just like what it comments up what the code did.
No, but what do the lines do? What are the lines doing?
You mean these?
Yes.
I thought it's just adding like the cell ID
What are you trying to do here? What's the point of the exercise?
to like do these
No, what are you trying to do? Why are you doing this? What is the goal of this?
to build the cells out.
You're trying to load an antimony model into cells, yes?
Yeah.
Fine. So you want to load it in once. You're
So
loading the same, you're loading it into each cell in multiple ways. That's not going to work.
so I just need one of them.
That's right. I would use the lines 58 and 59, get rid of the rest.
Okay, I got it.
Okay. Now, it's probably still going to fail for a variety of reasons here, but
Yeah.
okay. What happens when you try to run that?
It says like syntax error.
Okay, in what line?
Uh,
Do not switch back and forth until you know what your problem is. If you have a window open, don't move away from the window until you know what your problem is. Ever.
79.
It
But
won't work. It's line 55.
55.
Okay.
So do I? Oh, is
No,
it? No, not
no,
this one.
no. So it's important either use the twidit, either use the twidit, snippets, code snippets, so you don't have to remember the syntax, or else you have to learn basic Python.
Oh,
So
okay.
why don't you go to the code snippet under CC3D Python? It would be under SBML solver, under
XDML.
SBML solver, time step SBML models. Okay, that's the correct syntax.
Oh, I think I deleted the wrong one, but okay. Got it. That should
Okay,
work.
hit save.
Uh,
Okay, now you have a line 37 question. So let's go to line 37. Model
but
equals.
yeah.
Okay.
What's
So the
wrong?
issue here is indentation.
Oh, indentation. Should that work?
No, it won't.
Okay.
So in the first place, the antimony model has to be put at the top of the simulation.
Oh, okay, got it.
There we go. That should be okay. Except that there's one other thing. How does an equal sign work in Python?
It's like both are the
Model
same
equals
value.
nothing, and then you have a string declared.
Oh, but it's like in the slides you put 3.
You cannot have a carriage return after the equal sign.
Oh, okay. Do I just delete this or just model equals model?
No, just put the triple quotes after the equal sign.
Oh, okay. Like that.
Yes.
Okay.
Okay. So I do need you to learn basic Python syntax.
Okay, yeah, that works.
Okay. Okay, good. Okay, great. Anybody else? Kyle, how's it going? All right. So the next thing you need to do is to put the tracking fields in. And the tracking fields go after the init. And again, you can use the code snippets to give you the syntax for that. You'll create a tracking field for the value of A, a tracking field for the value B, and then in the start function, it's nice to create the dictionary entries for those. Okay. Give people a minute to do that. And then the last thing that you need to do is in the step function, you have to copy the value of A and B from the SBML dictionary to the cell dictionary. And here we're using a Python function. And this, if you've loaded this into every cell, you're not going to have an issue. But if you have loaded the SBML model only into some cells, then cell.sbml.myModel A won't be there. And so we will use the Python error trapping called try. If you use try, then if there is an error when you try to do the assignment, it will go to accept key error. In this case, we don't care, so we say pass. So this is a way of checking for whether cell.sbml.myModel exists or not. If it does, we copy it. If it doesn't, we ignore it. Okay. Again, if you've loaded the model into every cell, it's not going to be an issue. But in Santoshi's case, where she decided to load it into a single cell, into only one cell, you have to do this. That try function is the other place that you use try functions a lot is when you're doing file opening and closing, because it's possible a file won't be where you expect it to be, and you'd rather have your program give an error that is informative to the user rather than crashing. And so try is a functionality that's not available in every programming language, but it's quite powerful in Python. You can do rather a lot. There are a lot of options for tries, which you can look up. In our case, we don't need all of them. Okay, Emma, did you want to show us what you have, or do you want to?
So it's not exactly what we discussed in class.
Right.
So right now what I'm doing is just loading the model into every single cell. And that's it for now.
Great.
As a baseline, this is the type of sheet.
That should be fine.
So, you know, that's decent.
As you say, it's reasonably slow, but it's
Right, but I mean, this is workable, but look what happens now. And this is the part that's different from what we did, because I just tried to guess what to do. So I'm putting the cell dictionary entries in this way, and I'm actually referencing the fields.
oh, but so why? Okay, so you don't.
But look how much it slows down. I mean, look at that.
Yeah, it's pretty slow. I agree. Let's look back at what you're doing though. So you're going for cell in self.cellList.
Yep.
And then you're reading the value of the field.
Uh-huh.
A field and B field.
So what I realize is, do we have these chemicals represented in a way that's accessible both by the CompuCell and through the XML and by the model, or are they different objects?
Well, so there is no field. I'd say, so I'm not sure why you, so the first thing is I'm not sure why, I mean, what you've done is syntactically correct, but I'm not sure why you want to read the field.
What I assumed is, okay, if you make diffusion fields and define them with the same names as you reference in the SNBL model, they will interact. But now I realize that's not necessarily.
No, the SBML is completely segregated from the CompuCell except through those dictionary entries.
Okay, that's interesting. Now I realize. So I'll just delete this.
And so I mean, what you were trying to do is quite interesting because you might want to read back and forth. But so for example, suppose that we had virus in our environment and one of the input parameters to the network was the concentration of virus. Then you might very well want to do an uptake of the virus, or you might want to read the concentration of virus in a field and pass it to the SPML model. So what you're trying to do there is absolutely a reasonable thing to do. I'm not sure why the access to the field is. I think the access to the field is slow because I don't understand why the access to the field is slow there. My guess is that somehow you're trying to, it's pulling the whole field in and then finding a value inside of it each time. It shouldn't be doing that, but that's the only thing I can think of for that one.
So actually now I'm confused because in the call to track cell level scalar attribute,
Yeah.
we have to specify the field name.
Right. So
Is-
that field name should not be the same as a chemical field.
Okay, but if it's a field that only exists in the SPML model, CompuCell will get that reference or not?
Ah, okay. So Emma asks if there's a name in the SPML model that happens to be the same as the name of something else existing in the CompuCell Python or XML, what happens? And the answer is that the namespace for the SPML models is separate.
So
And so it would be confusing to name. So the variables in the SPML are not fields, they're scalar values.
okay,
And they live inside the cell. Because networks are not spatial objects.
yeah. Yeah.
So if you create a field named A and a network variable named A, those are unrelated to each other. The only reason we're invoking fields is so that we can display the value inside the cell.
Okay, so when we make this call to track cell level scalar attribute,
Right.
we're actually creating a new field named A.
It's creating a field named A, whose job it is to say that within the spatial domain of this cell, show a color that corresponds to the value of that dictionary entry. In the old days, we would have had to do something elaborate because if we wanted to display a scalar value, which was associated with each cell, we would have had to figure out how do I show that. And so tracking fields are designed to show scalar values inside of cells. That's a great question. Okay. So, okay. So are other people, how about Santoshi and William? Is this working for people? Santoshi, William, were you able to create the tracking field? Okay, William?
Yeah, I got it.
Okay. Okay. All right, so you should see something that looks like this. In the case of Santoshi's one where she loaded it into a single cell, it should look like that top row. You get something that looks like that where there's one cell that's colored and the rest are not. Okay, we didn't do by cell type. That's something you could do as a homework assignment. And if you load it into all the cells have exactly the same model in them. And so you should get a uniform color in all the cells. Is anybody having a problem with that, or did that work for people? And again, I think it's important to take the time with this because everything we do is going to be based on the ability to load things into cells and run them. All right. So now that you've done that, your next assignment is change the rate constant in each cell, the K1, to be its original value times a random number between 0.3 and 3. And do that in the start function. So what do you have to figure out how to do here? First thing you have to do is remember how to generate a random number. And remember that you get numpy as NP in CompuCell. So you may have to remember, how do I pull a random number between 0.3 and 3? Then you have to remember how to reference the parameter value through the dictionary. And you'll loop over each cell, and you'll change the value of K1 by multiplying it by a random number between 0.3 and 3. Okay. And now run it. And the question is, are the colors for your tracking fields now the same for every cell or not? Santoshi, you're going to need to load your model into every cell. So you're going to have to switch your code, the one line of code where you did the load, you'll have to load it into all the cells, not into just the single cell number 20 for this to work. So Emma asked, can I explain about the dictionary? You're in your start function, and okay, fine. And so now you've added your antimony model, and now you're going to pull a random number. That's perfect. And so now what you need to do is you need to read the value of the, okay, good. So now if you go to twit cc3d Python, and go to SPML and just go to, boy, there are too many options here. Get value for SPML in specific cell. There we go. Okay, good. So now you want to go cell.spml dot whatever you called your model, test model. Okay.
It's just a fix.
And then K1. And now you'll have to say basically times equals factor. Why don't you try running that and let's see, make sure it works.
Oh, because it crashed. Okay.
Let's look at let's look at the tracking field. And
Yeah.
you see now they're different colors. Because you've changed the rate of reaction. They're not doing anything. Those equations are still not doing anything, and they're not responding to anything that you're setting in the simulation, except inside the model. But you can now have effectively you're running a different network inside each cell.
And I kind of coded out the random thing for
Okay,
this
you want to show us
proposal.
what you got?
Yeah, but it doesn't really work because I coded over here. These. What is it type?
So there's no model replace. You don't need any of that.
Okay. So will these be fine? Like just random 0.323.
So what you need to do, let's just change K1. You don't need to change K2.
Okay.
Nothing wrong with changing K1, but take two too, but okay. So when you load the model,
Yeah.
the parameter values, put your cursor down in line 31. You can't do anything till after you've loaded the model.
Okay.
In line 31, go to the code snippets, CC3D Python,
Mm-hmm.
SPML solver, and then no, not time step. There, get value for get value for SPML in specific cell. There you go. Okay. That
That's
references now what you need. So now you have to replace model name with the name of the model. No,
model
model
name with
name.
you mean this one?
Yes.
Oh, okay.
Well, right. And value name is now K1,
Okay,
the
gotcha.
name of the parameter. And now you want to multiply that by K1 factor. So times equals K1
You
factor.
mean this times or just K1?
No, the value of, so you now have the value, you're now referencing a variable, which is K1
Yeah.
inside the cell. So you
Uh-huh.
now have to multiply that by, no.
So not this one?
no, that's the name of the variable.
Yeah.
If you
So
multiply by the name of the variable by a number, you're going to get something that's meaningless.
do I just K1 times
No.
3?
What
No.
is a dictionary? What is a Python dictionary? So a dictionary entry is referenced by what's called a key, which is an alphanumeric string.
Uh-huh.
So cell.spml.mymodel is a dictionary,
Yeah.
and
So if I type,
K1 is the name of a variable
yeah.
inside of that dictionary.
Uh-huh.
And now you want to multiply the value of that dictionary entry by K1 underscore factor.
Okay.
So you
So,
would type cell.spml.mymodel K1. You don't need to type it again.
oh,
Just
it's copied.
times, no, don't change anything.
Oh.
No. At the end of the line 33,
Mm-hmm.
if I have a variable x and I want to multiply x by 1.1, what do I type?
Times 1.1.
But just type in line 34, type x. I have variable x, and I want x to be replaced by x times 1.1. What do I type?
Oh.
No.
Mm.
So I could type x times 1.1. That would be all right.
Okay. I don't quite.
But that's not, you need x equals x times 1.1.
So it's just divided by 1.1 then.
No, type x equals x times 1.1. Okay, that's legitimate. So you could type that, you could use that form in the line before. You could say cell.spml.mymodel K1
Uh-huh.
equals cell.spml.mymodel K1 again
Times 1.
times K1 underscore factor.
So you want to multiply, you just star multiply 1.
In this case, K1 underscore factor, which is what you type.
Like that.
No?
Yes.
There you go. That's right now. Yeah. Now get rid of line 34. Okay, try running that.
Line 30.
No. Save it. Always use the save on the right. That's the one with the 2. Yeah. Okay.
Yeah. It's
Okay, then close player and run it again. And open it again. Line 30.
yeah.
Let's see. Where is the modify? Oh, I see. You changed the name. Scroll up, please. Why does line 30 say modified model? Well, you don't have a model string called modified model.
Oh,
There's
so
only
I
a string
just need
called
to.
model.
Okay,
So
that worked.
Try that.
Yeah, this is what I got when I first typed that.
All right, so that's a syntax error in your antimony.
You
So let's
mean,
look at your antimony.
but I didn't change anything about it.
Yes, in line 8.
Line 8?
Yes. Okay, now run it.
Okay.
So you do have to antimony. No programming language will forgive you if you type a character that's not the right character. So if you type an arrow instead of a dash, it won't work.
Okay.
So you actually have to have the right characters.
Okay.
That's not a CompuCell issue or even a Python issue. That's just a programming issue. Maybe nowadays with current uh, IDEs, it'll actually tell you what it thinks your mistake was. But a classic one, it doesn't do that. So it's not good, I guess. Okay, fine. So why don't you display the field, the tracking field?
You mean over here?
Yes. Show instead of cell field.
But I only have cell field. I
So
don't
you
know
didn't create the tracking fields.
what? I
You
thought
didn't create the tracking fields.
the tracking fields is this one.
So why are these in defs? Why do you have def init and def start here?
Which line are you talking about?
Line 60 through 84. So you have to put the code that you've got in line 60 down through the end in the appropriate places in your
Oh, okay.
simulation.
Gotcha. I'll just
So you can't have two def starts. You have your start. There you go. Right. And you already have that, right? You've now got this twice.
Oh, okay. Wait.
You don't need that at all. So what you
Okay.
have is, okay,
But I need this one, right?
that needs to go in your step function. And the indentation has got to be fixed
Okay.
in line 45. Okay. All right. We'll do it on the screen now.
Okay, yeah.
Move line 69 and 70. Cut those. And those go after the init there. Yes, there. There you go. Okay.
Do I still need these? I thought we only need the ones below, right?
You need them in lines 39 and 40. You don't need to do this for cell. You've already got it.
Okay.
But you need them.
Like behind it?
No, you don't need line 37
Okay.
because you already have the iterator in line 28.
Gotcha.
But the indentation is wrong. Okay, good. Okay. Get rid of line 65 through 73. Those will cause errors.
Okay.
And then now line 66 through the end have to go into your step function. No, they go after the. Get rid of lines 51, 52, 53, which don't do anything. And fix your indentation. Okay.
Okay, yeah.
Okay. So now that you have a model that has cells where you can load a model into individual cells, and now that you can read and write things from the model to Python CompuCell Python and back, we're ready to actually build a model that does something. So what we're going to talk about now is delta notch patterning. And delta notch patterning is a very fundamental biological mechanism. It uses what's called contact inhibition. So if a cell has high delta, it tries to make its neighbors have low delta. If a cell has low delta, it tries to make its neighbors have high delta. And this gives rise to salt and pepper patterns. We sometimes call them checkerboards, but actually it's not quite a checkerboard. It's a checkerboard where if you imagine a checkerboard where you had one black square surrounded by 9 red squares, or 8 red squares, that would be the kind of checkerboard we're talking about. Okay, and you can see this example in the intestinal crypt, where you have alternating cell types. You can see it in the Drosophila inner ear, where you have alternating cell types. You can see it in the eye of the zebrafish, where the photoreceptors are defined by delta notch signaling. Delta notch signaling also is very important in neuronal pathfinding, and it's also very important in vascular development. So when cells are going, when you have cells migrating in a stream, the leader cell of that group of cells often has a different state from the cells that are in the stream. And the cells that are the way the cell determine who's the leader is through delta notch signaling. For example, neural-crest cell migration, that happens. In vascular development, when you have a branching morphogenesis, where you have a blood vessel and the cells grow out of it, the leader cell and the follower cells have different delta notch states. And so delta notch is probably the most important contact-based signaling mechanism where you get differences of cell type. There are a lot of mechanisms where you say to your neighbors, I want you to be the same type that I am. But this is something where you're saying to your neighbors, I want you to be different cell type from me. And it's a mechanism that's used again and again in developmental biology, and also in repair, tissue repair. And failures of delta notch can be related to cancer as well, and a lot of other developmental problems. And so the basic idea here, if we look at this little biological diagram, is there are actually 2 separate receptors. They're both transmembrane receptors. One is called delta and one is notch. And they're what are called heterophilic receptors. That is, delta on cell 1 binds to notch on cell 2. Notch on cell 1 binds to delta on cell 2. Okay. When delta binds to notch, it sends a signal from notch that inhibits cell specialization. So it keeps the cell in a more primitive state. It also promotes proliferation. And it also inhibits the production of delta. So active notch inhibits the production of delta. And so if you start out with equal amounts of delta and notch on the same cell, the cell will tend to move into a state where you either get a lot of delta or a lot of notch. And there actually is some hybridization where what's called having 2 receptors on the same cell is called cis, and 2 receptors on touching cells is called trans. And so delta and notch in trans send a signal. Delta and notch in cis on the same cell tend to wipe each other out. They tend to net each other out. We're not going to do that particular detail here. If you look, for example, at Oriolon's books, you'll find very detailed models of very complex models of delta notch. Lorenzo Veschini has done a lot of work on more complex delta notch models. We're going to do the absolute minimal possible delta notch model here today. Okay, but when notch is activated, it inhibits delta in the cell, And that's the thing that we're going to model. And so the result is initially all the cells are about the same. They all have a little bit of delta and a little bit of notch. The ones that have high delta will inhibit delta in their neighbors. And so you'll get high delta. And depending on the model, you'll either have delta and notch in phase with each other. So high delta and high notch in the same cell, low delta and low notch in the same cell. Or more typically, high delta, low notch versus high notch, low delta. That depends a little bit on how you build the models, okay? So this is a contact inhibition mechanism. If I have a lot of delta, I tell my neighbors have low delta. There's a very simple model of this due to Collier.
Can you say that again? Just that last statement. If I have a high level, I tell.
If I have a high level of delta in my cell, I tell my neighboring cells, I want you to have a low level of delta.
So it's signaling somehow.
So this is a contact signaling mechanism. This is literally a contact signaling mechanism. The cells have molecules that stick out of the cell. They bind to the molecules that stick out on the neighboring cell. And when they touch each other, they send a signal.
Okay, thank you for explaining
Great.
that. So,
okay, and so that's diagrammatically shown here in a very simple way. When notch is inactive, it promotes active delta. When notch is active, it inhibits delta. Now, in reality, there are a bunch of steps, not just one step. But we're going to use this Collier model, which has the correct general behavior and only has two equations.
I'm sorry to ask one more question. So it looks like from the diagram, the notch is the receptor or something. It's capturing.
They're both receptors. They're both
They're both receptors. Okay. So it goes.
Although in this case, we are only looking at we're thinking of delta as sending a signal and notch as receiving it.
Okay.
In reality, they are both receptors. Delta does actually receive signals as well as send them. But we're thinking about this here as delta. If we think about sort of a male and a female plug, you can think of delta as being, well, actually the signal comes the other way. So if we think about a wall outlet, the signal is coming from the wall and going into the cord, right? So,
sorry for getting us off track.
No, you're not off track at all. It's a bit tricky to think about these, and if I'm not clear, I definitely want to make sure that the basic biological idea is clear. So when we have active delta, it will send us, and it connects to a signal cell in the neighboring cell. It will send a notch. It will activate notch in the neighboring cell. When notch is activated, it will turn off delta in that cell. When delta is inactive in that cell, it inactivates notch in the neighboring cell. So we can't really understand this process looking at only one cell. This is a classic example of why no matter how much detail you have of what's going on inside one cell, you can't understand the system. Because the circuit only makes sense if you have two cells that are interacting with each other. If we just have delta and a notch within the cell, it doesn't tell us anything. And I think that's perhaps where why it's a little bit confusing. We're used to thinking about the network inside a single cell. In this case, we really have to track it through two cells, at least, to be able to understand what's going on.
So, okay, so part of it you said is spatial, and that if it's got the other thing around it, it signals the other cell.
Right.
And then is there any timing thing to this too, or is that kind of constant, or just
so let me walk you through the equations of a Collier model, which are exactly answering that question. It's perfect.
Okay.
You couldn't have set up what I was going to say next better, Kyle.
Okay, thanks.
Okay. So what we want to know is how much delta do we have in our cell? And the amount of delta that we produce is a constant divided by the amount of notch. So if we have no notch, we produce delta at a rate nu. And as we have more notch, we produce less delta. That is, notch activated inhibits delta. So that's the first term here. And this functional form 1 over 1 plus b times n to the h is what's called an inhibitory Hill function. That's a sigmoidal function that says basically if we have a lot of notch, turn off the signal. When we create something, when we create a chemical, we always have to have it decay. And so we have what's called first order decay kinetics. We lose a certain amount of delta per time step. So if we had no notch, this would read d by dt equals nu times 1, constant rate of production of delta, minus nu times delta, the constant rate of decay. And that will give us a steady state where the concentration of D is 1. So the equilibrium concentration of D within the absence of N is 1. Now what happens with notch? Notch is produced not by delta in our own cell. It's produced by delta in the neighboring cell, delta active. And so here, dN by dt is equal to delta, not delta in our own cell, but delta in the neighboring cells. And in this case, the more delta our neighbors have, the more notch I have in my cell. This is what's called an activating Hill function. If my neighbors have no delta, I produce no notch. If my neighbors have a lot of delta, this delta neighbor to the k over alpha plus delta neighbor to the k, the maximum value of that is 1. So if my neighbors have a lot of delta, then the rate of production of notch is 1. And I have decay of notch at a constant rate. And so if delta in my neighbors is very high, my value of notch is 1. If my value of notch is 1, that knocks down my value of delta. And so I'm going to get a seesaw. Now, the exact parameter values matter a little bit. In this case, we're going to use A equals 0.01, B equals 100. And that scaling, we could have rescaled everything to be 1, but that's what Collier used. We're going to use an exponent of 2, which is fairly smooth for Hill functions. And that's the set of equations that we're going to run. So, again, the basic idea is that when a cell has its neighbors have a high level of delta, it stops producing notch. When a cell has neighbors have low levels of delta, it produces notch. So it's a switch. And that'll produce this hexagonal pattern. Okay.
So is this primarily genetically regulated or physics, chemistry related, or both? Maybe it is both.
So if I were to give you the whole delta notch circuit, the production of delta and notch is a gene regulatory network. However, the things on top of them are signaling networks.
Okay.
So this is actually, the real system is a combination of signaling networks and gene regulatory networks.
So it
And we've
gets
simplified
complicated.
all of that into two numbers.
Okay, get it. I got it. It's just, it's complicated, and this is a way to mask some of that for now.
Well, so one of the things that happens is, for example, when delta binds to notch on the surface of the cell, the notch molecules actually split in two and destroyed. And the intracellular component of notch is then released, goes into the cytoplasm, and eventually then sends a signal to the nucleus to to affect the delta production.
Wow.
And so we're getting rid of all that chain of signaling and just having it be one equation.
Got it.
But it's not a bad, actually this is a pretty good approximation. If you compare this to more detailed models, it works pretty well. Okay. So based on what we just did, and we're going to probably take a break in a few minutes because we're a little over our break time. I'd like you to create a 42 by 42 simulation. You can make it bigger or smaller as you like. We only have one cell type. Use volume local flex and a contact energy. Use uniform initializer to fill the whole simulation with cells that are initially 7 by 7. And then in the start function, you'll have to say that the target volume of each cell is 49 and lambda volume is 2. So we've done that before. Okay. Then you're going to create an antimony string. Well, you could start with the simulation that you just had and change it in simple ways to do this. Write an antimony string that corresponds to the 2 ODEs that I've written here for delta and notch. Load that model into each cell. Make the initial values of delta and notch random numbers between 0.9 and 1. So you're going to make them almost 1, but not quite, with random values. And why don't we focus on that as your initial task? And then the thing that's going to be a little bit more work, and I'll walk you through these, is going to be calculating the D average parameter for each cell. So I have to then iterate over my neighbors of each cell, read the value of D for each one, and average that. And then I'll create auxiliary fields to display the values of delta and notch. The only thing that we are doing here that we did not do in the example that we just did, 13.2, the only thing that's different is that we are going to read the values of our neighbors and average. So everything you need for this model was done in the simulation that we just did in 13.2, the one we just finished. Okay? So again, to walk through this, create a space filled by cells using uniform initializer, type the model in antimony, add it to each cell in the start function. In the step function, loop over each cell. For each cell, loop over the neighbors of the cell. Read the value of D for each neighboring cell, average it, and then overwrite the value of D average in my cell with the neighboring values and visualize the pattern. Okay, and you'll see that when it works. So let's go through that. First thing is to create a 42 by 42 simulation domain with one type of cell. And you're going to do this in Wizard. Start out, again, create a 42 by 42 simulation, use Wizard. One type of cell, volume local flex, contact energy always. Use uniform initializer and fill the whole domain with cells of size 7 by 7. And of course, you'll have to assign a target volume of 5 and 11. I don't know why it says target volume of 5. It should be 50. The steppables file, it should say assign target volume of 50 and a lambda volume of 2 to each cell. Okay? And give me a thumbs up when you've got that working. That's step 1. Step 2, and here's how you implement that if you have trouble. So we're using uniform initializer and self.cell list, target volume 50, cell lambda volume 2. And what I want people to do is after each step, run it, make sure it works. So that you've got, if there's a problem, you catch. Okay, the next thing I want you to do is write the antimony model. And we know how to write an antibody model. It's somewhere at the top of the simulation. We're going to say model string or model equals, triple quotes, and then the string. And I'm not going to give you the exact syntax, although I can do that in a minute, but you're going to be typing d by dt equals new times 1 over 1 plus b times n raised to the h power minus d, and dn by dt equals d to the k. And this is a different d. So d average and d have to have different names. You can call one d and d external, for example, over a plus d to the k minus n. I think you got the parameter values here. And I'm just reminding you of the syntax you need. If you write J1 colon arrow goes to D, that's equivalent to writing dd by dt. Then you write the semicolon and whatever math is on the right. So you'd have two lines. J1 arrow goes to D, J2 arrow goes to N. And reminding you that the exponentiation is a caret rather than a star star. And you have to give the values of A, B, new, and I have K and H potentially being different, but here they're the same. So at the moment, Emma asks, how do you implement the D average? So at the moment, we're just going to have that be a parameter, which we can call D average or D external, which means you're going to have to give it an initial value inside of the model. And then what we're going to do is we're going to overwrite it. At each time step, we're going to read the value of our neighbors and supply that to the simulation inside. So that's the only thing that's new from the previous simulation that we just did. This is what it looks like when I did it. There are a lot of different ways you could write the same thing. I mean, it'll work. Putting the model string in and not doing anything with it shouldn't crash, but always, once you've typed something, run your simulation. Make sure you didn't introduce an indentation error or have the William had the issue where he put a carriage return between the equal sign and the triple quotes, which won't work in Python. So it's always good to, every time you add a little component to your model, check that it runs in player before you keep going. I mean, you could put it inside. I said to put it on a square lattice with one cell type. You could use the simulation with blob initializer. It'll work just fine to do that. In fact, one of the later exercises is to try different spatial configurations of cells and see how they behave. So the next thing we have to do is load the antimony model into all the cells. And we'll do that in the start function. And here I've loaded it, instead of running a loop over all the cells, I've loaded it into all cells of, there's only one cell type, I've loaded into all cells of that cell type. And the one thing that's a little bit different here is that I've made the step size 0.2 because I know in advance what the time conversion has to be. If you want to do a loop over all cells and add it that way, that's fine too. It's up to you to decide how you want to load it. But you do need the model in every cell. Again, if you'd rather use the code from before where we looped over the cells, that's fine too. This is just showing you a different way to load the models. Of course, if you called your cell type something different, you have to use whatever that type name is. If you called your model something different, you have to use that. Now, the next thing we have to do is randomize those initial values of D and N, which is why we did the little randomization exercise in the previous one. So again, remember that we're going to reference the values of D and N, cell.sbml.dn, if that's what we called the model, D and cell.sbml.dn N. And we're going to make those equal to np.random.uniform 0.9, 1. And we'll do that in the start function. And then, as usual, in the step function, we'll do that time step SPML model. So add that as well. And again, check that it works. Emma says the pattern forms and disappears, which is right, because we haven't coupled the cells. Okay, this is the result for that. Here's some code for it. We add the model, we iterate over the cells, we make each value of D and N uniform random between 0.9 and 1. In the step function, we time step them all. Okay, the next step is the one new step. This goes in the step function. And the algorithm is this. We loop over all cells. We create a placeholder value for the average. Total equals 0. We now iterate over the neighbors of our current cell. We check if neighbor to make sure there's no medium. In the case of filling the whole lattice, there's no medium, but you might have a different configuration where there's medium. If neighbor, we read the value of delta from the neighbor. We add that value to our placeholder. We keep track of the number of neighbors. When we've done with our loop, we divide the total by the number of neighbors. You better check that the number of neighbors isn't 0, so you don't divide by 0. And then we overwrite the value in our current cell of D average with that average read from the neighbors. Is that clear? And if you get ahead of things and you want to do something that's a little more biologically realistic, you can keep track of the contact area with each neighbor, weight the signal from each neighbor by the contact area, and then instead of dividing by the total number of neighbors, you divide by the total area of all the neighbors. That's a little bit more biorealistic. It doesn't make a huge difference to the result, but if you want to actually model the biology more accurately, that's. Again, if you don't remember how to loop over all cells, go to Visit in the Twidit code snippets. If you don't remember how to iterate over neighbors, go to Iterate over neighbors in Visit in code snippets. And I've got the here in the line, how you reference the value of D in the neighbor, neighbor.spml.dn D. Now, if one wanted to be more Pythonic, one could do something like, instead of doing the loop, the average by hand, you could stack. You could create a list in which you put the value of D from the neighbor, and then you could use a NumPy function to get the average value. So here's one way to do it. Not necessarily the most elegant way, but one way to do it. So in the step function, I iterate over the cells in my cell list. I set D average to 0. I set the number of neighbors to 0. I iterate over the neighbors for my cell. If my neighbor is not medium, I add the value of D to D average and increment the number of neighbors by 1. When I exit the loop, if there are any neighbors at all, I set cell.sbml.dn D average equals D average over NN. Probably if there are no neighbors, I should set D average to 0. Okay, Emma's trying to go to the advanced problem, which is, she says, how do you get the shared boundary area? Actually, when you do the iterator, common surface area is the shared boundary area.
Oops, I made that underscore.
And so if I wanted to, I could just, instead of saying NN plus equals 1, I could say NA plus equals common surface area. And if I did that, then I would wait instead of d average plus equals neighbor, I'd do d average plus equals common surface area times neighbor. And then I do the same division at the end. So it's not that big a deal to add that feature. Something here that maybe is not so familiar, and I've got it in parentheses, which actually is a bit of an issue, is that iterators, and we're used to iterators in a language like C, well, like C or Fortran. Iterators are over a variable. In Python, you can iterate over complex objects. And so in this case, the iterator over get neighbor data list actually returns a tuple. It returns neighbor and common surface area together. It always looks a little funny to me when you have two things on the the left-hand side of an equal sign. X comma Y equals something looks strange because in a lot of languages that doesn't work. Python it does. So now the last thing you have to do is add a tracking field to track the values of delta and notch. And now the last thing we have to do is create the tracking fields to display the values of delta and notch. And we have to copy the values of cell.sp_dn n to cell.dict of n, and same for d. So again, the only thing that's new from the exercise 13.2 is this issue of iterating over the neighbors and getting the average. Everything else is the same. So here's a reminder of how to create the tracking fields and how to do the copy. So that's the result there. This demo is available in the demos folder for this class. You should always look at those in advance because almost everything is done out in those.
Gotcha.
So you should download them before the class. And I get something that looks like that. Alex and Emma, do you want to try letting us see what you got? Okay, that looks not crazy. Your cells are fluctuating a lot, which is okay. They're moving around a bit. So you put in a surface area constraint. Trying to think if we could do something to make them fluctuate a little bit less. I'm a little puzzled about why they're fluctuating so much, but okay, let's look at your code. This looks, I mean, it doesn't look crazy, but let's just make sure everything is. So there's your model. You've got tracking fields for D and N. You called the model DN. You added it to all cells. And you called mean D is the one that, yeah, okay, fine, good. Okay, and now you're going to set initial values there. Set them uniform random. Yeah, we don't need to set them equal to 0 if we're going to set them uniform random. It doesn't matter. Okay. And now we're going to do the cell coupling. Whoops. And then we're going to do the cell coupling. You say delta is, okay, so you're doing an append. Okay. Oh, So where are you getting cell surface from?
Oh, I just thought I would normalize, so it's the proportion.
Okay, so I do not know that cell dot surface will return the sum of all of the numbers that are returned when you add everything up. I agree that it ought to, but I'm not sure that it does. So I would check that. In other words, if you're going to do that, I mean, I understand why you want to do it that way, but I would yeah, boundary area, yeah. Then when you do the average, you want to divide by the sum of the. Yeah. There you go.
Is it 2 or 3 in Python? I always forget.
For comparison, 2.
Which languages? Yeah,
I don't know. I've never seen 3.
maybe JavaScript?
Probably. JavaScript always is funny too.
Yeah.
All right.
Yeah, it's working.
Okay, so then it should be all right. All right. You trust our code more than I do. Okay, good. So then it should be right. The only thing I might do is, since the cells are fluctuating so much, maybe make the lambda volume in the start function a little bigger, so they don't move around quite so much. Make it 7 or something.
The lambda surface.
That's probably what it is. 'Cause I wouldn't use lambda surface. You don't need it. You don't need the surface energy.
Surface, then I can't actually call this.
Oh, so okay, I see why. Okay, that's why.
That-
So what did you change? Just the volume constraint?
Surface, but I don't know what the. I don't really want to do this. Gosh.
So you know what you can do? Make cell dot lambda surface 0. Then it'll still return the value, but it won't apply the energy function. There you go. That's better. So what was happening, I think, was it was trying to force it to a large surface area. Yeah. They're still fluctuating a bit much. So I would make, again, I'd make lambda volume a little bigger. So make it 5. Yeah, there we go. There, that's better. And let's see what we get. So the pattern will not necessarily, when things are moving, you won't necessarily get a nice salt and pepper, because it's possible that you don't have, there is no pattern that satisfies everything. Right? If you have an even number of cells and you want to have an alternating one, you're okay. But if you had an odd number of cells, you're always going to have a mismatch. So you can have what's called frustration in the pattern. But this is working pretty well. You see isolated high values of delta surrounded by blue, and then some intermediate ones where there's a conflict between the values. So that looks great. How about Alex? So your cells aren't moving as much. And so it's staying a little bit more, it's a little closer to the pattern that I got. Kyle, were you able to get it to work? Again, if you can't get this to work when you're writing it, download the code, the online code. Okay, the last thing I want to do today, we're not going to get to the two other examples, which I had hoped to do, which are very similar to the ones we just did. So there may be homework. I want to talk about steering panels. So CompuCell was not originally thought of as something that was going to be distributed as packaged models. And it was when we started using CompuCell on NanoHub that this really became an issue. But if you're going to write simulations that are not things that you're running for research, but you're going to write simulations that you're going to have other people use, you don't want to have them edit the code to change parameter values. And we have that steering panel, which allows you to change the values of things that are specified in the XML, but it doesn't let you change the values of things that are specified in the Python. And so steering panels are a way of creating not the most sophisticated, but a basic user interface where the user can change parameter values, any parameter values you choose to make available to them. And I have the comment that it's especially useful for NanoHub applications, but anything which you want to distribute, for example, is an educational application where the user is going to be changing parameters. You'll want to be able to do this. And as usual, the Twidit has the code snippets built in. Now, steering is what's called event-driven, and event-driven programs are not something that we always do. And so this may be a little bit unfamiliar to people. I'm sure some people in the room have done events before. But what we have to do when we're going to create a steering panel is first we create a steering panel window, which defines the parameters of variables we're going to change. In Player, the steering panel that's built into Player is predefined based on the XML. This is now going to be a user-defined one. Then we have to define a function called a callback that watches for panel updates and reports them. And then we actually have to change the values of the updated parameters. Because this is going to be a function that doesn't run all the time. It only runs when somebody actually goes and touches the steering panel. Okay. Has everybody done that before? So it does take a little bit of getting used to, but once you've done it once, it's not so bad. Okay. So CompuCell only lets you have two kinds of steering options. It has if we had IPy widgets, you'd have lots of them, but we only have two. Maybe we should add more. So we have a function which goes in between the init and the start function, the same place we do our tracking field definitions. We include a function which is called def add steering panel. And within that steering panel definition, we define the name of the variable that we're going to change, its initial value, its default value, the minimum allowed value, the maximum allowed value, how many digits we're going to return. Is it going to return 1, 2, 3, 4? Is it going to return 0.1, 0.2, 0.3, 0.4? Is it going to return 0.01, 0.02, 0.03, 0.04? And this is a slider. So this is going to give us effectively a roughly continuous value. Okay. So that's one kind of value. Okay. The next thing that we have to do is we have to write a function that notices when there's been a change in the steering panel. And in this case, we have a function called process steering panel data, which has to be done after we create the steering panel. And in this case, we're going to read the value from the steering panel. Self.get steering param and the variable name. Now, if that's all we do, when we exit the function definition, when we exit the function definition, we lose those values of A and B. Those values of A and B are local to the function definition. And so we typically are going to have to write the values, overwrite the values in that function so that they get out, so it does something. Okay. Another thing that you can do, which can be useful, is if you want the simulation, this is again more for an application than for the kinds of things we're going to be doing here. But suppose that you want the person running the simulation to have to choose something at the beginning of the simulation. In that case, you could have a simulation that depends on a steering panel entry that says select simulation to run. And you could have a feudal loop that says while steering panel entry simulation type is select simulation to run. That means the user didn't select one. You just sit there and keep looping. Once the user selects the simulation, you set the simulation type to whatever they did, and then you start running the simulation. You could also do this in the opposite direction. You could have something where there was something, a slider that said pause, and if the person slides it to pause, then you enter a feudal loop and wait. And so you can actually use the slider like the pause button or the start button in the simulation. But this gives you a lot more control because you can say that the user has to define the values of certain parameters first before the simulation starts. So what we'd like to do now, and here it says use the code for the previous exercise, which we didn't do. So maybe I have to be a little bit careful here. And I'll show you the code so you don't have to invent it for yourself. We're going to add steering panels to change the values of A and B in the simulation. Remember that in our delta notch, we had A and B. A and B set the scale at which delta is inhibited by, which notch is inhibited by delta. A sets that scale. And B sets the scale, I got exactly backwards. A sets the scale at which delta turns notch on, and B sets the scale at which notch turns delta off. And if we change those, the color patterns that we've got will change. We know that the initial values of A were 0.01, and the initial value of B was 100, so we know approximately the range over which we need to do that. And again, if you're stuck, we could use the exercise 4.1.2 that I've saved. And so here's what we want to do. We're going to first create an add steering panel. And this goes in your code just after you've created your tracking fields. And so why don't you try that? And you can just follow along with the code that's on the screen. Again, there should be Twidit pull-down menus that give you this code. So you don't have to type all of it. We're going to add a steering parameter panel for A. That's a slider. and a steering parameter for B, that's a slider. And we'll let the value of A go between 0.01 and 2, 0.02. We'll let the value of B go between 100 and 200. Sorry, between 0 and 2. If you put this code in, just this code, and not the rest of the steering panel code, when you run it, you should pop up a window that has A and B with a slider, but moving them won't do anything because we haven't written the callback in. But let's make sure that that works. So the next thing we have to do is actually take action based on what we've typed. And so this is now a function which is called process steering panel data. And we have to read the new value of A and B from the slider. And then we have to iterate over our cells and update the values of A and B for the cell. And so if you add this code, now when you change the values of A and B, you should see the pattern change. So see if we can get that to work. And now you've got something that you could actually use to teach. Not teaching CompuCell, but teaching biology. Because you could have the simulation run for students, they run it, and you tell them, if I change the strength of delta inhibition by changing A, what happens to the pattern? If I change the strength of notch activation by changing B, what happens to the pattern? Does that work, Emma? Okay, so if we now pull the slider, does it change the pattern? Yes, it does, I think.
But I have.
Okay. Well, all right. So that's where we'll stop, and your homework will then be to, for the week, we'll give you a homework assignment on this. Get that up to speed, and maybe one of the other exercises that I'd planned for today will show up as a homework assignment. I realize the sliders are take a little bit of getting used to, and we've talked about slider. There's also a pull down menu.